Hey guys, welcome back. This time around we're going to finish our discussion in biochemistry by looking at the last of the organic molecules. So far we've looked at carbohydrates, lipids, and proteins. This time around we're going to look at nucleic acids. And nucleic acids are going to be composed of carbon, oxygen, hydrogen, nitrogen, and phosphorus. They're going to have structural units, monomers, known as nucleotides. Now, each one of these nucleotides is going to be composed of a nitrogen-containing base, along with a pentose sugar and a phosphate group. Now, what makes each of the nucleotides unique is going to be what type of nitrogenous base it has. There's 5 types of nucleotides listed. They're going to be adenine, guanine, cytosine, thymine, and uracil. And as you can see, we typically describe these when we're discussing them by the first initial of their name. Now notice what's unique about these. Not only do they have a different type of nitrogenous base, but 2 of those nitrogenous bases are 2 ring structures, known as purines, the adenine and the guanine, whereas the other 3 are going to be only a 1 ring nitrogenous base, in the case of thymine, cytosine, and uracil. Now there are 2 classes of nucleic acids. There's DNA and there's RNA. DNA stands for deoxyribonucleic acid. The D in DNA comes from the pentose sugar, the deoxyribose sugar, that is part of each of the nucleotides that are going to be found in the DNA molecule. DNA is going to be a double-stranded helical molecule. It's going to be found in the nucleus of cells as well as the mitochondria. Its role is going to be to guide us through our genetic blueprint. Now specifically, the genetic blueprint codes for how to build a protein in each and every protein found in our body. Now DNA will replicate itself before the cell goes through division, ensuring that we have genetic continuity. It provides and only provides the instructions for protein synthesis. If we're looking at a DNA molecule, we would find it's composed of 4 different types of nucleotides: adenine, guanine, cytosine, and thymine. Notice uracil is not found in a DNA molecule. Now also notice that each of these nucleotides are going to have the deoxyribose sugar as part of its backbone. And when you look at DNA molecules, as I said, it's double-stranded. And it's double-stranded not haphazardly, but it's double-stranded into a corkscrew, actually a counterclockwise corkscrew. And it's based upon 2 parallel strands of DNA molecules that are interlocked based upon a complementarity. This complementarity is anywhere there is an adenine on one strand, there's going to be a thymine on the other. Wherever there is a guanine on one strand, there's going to be a cytosine on the other. And notice this interaction is a hydrogen bonding interaction. In fact, there's a double bond interaction between adenine and thymine, and there's a triple bond interaction between cytosine and guanine, wherever it happens to be found. In fact, we could do a DNA replication fairly easily. If I know what one strand of DNA is, say it's A, C, G, C, then I could automatically know what the other strand is going to have there. Because anywhere there's an A, on the other strand there's a T. Anywhere there's a C, there's going to be a G on the other. Now the other type of nucleic acid that we have is going to be ribonucleic acid. Ribonucleic acid, or RNA, is going to be unique from DNA in several ways. One, its pentose sugar is not going to be deoxyribose, but instead it's going to be the ribose sugar. Another way it's unique is instead of thymine as a nucleotide, it's going to have uracil. Now the adenine, the guanine, and the cytosine are also found in RNA molecules. It's just that, remember, the pentose sugar for adenine is going to be a ribose if it's going to be found, if that nucleotide is going to be found in an RNA molecule. RNA molecules are single-stranded molecules. They are found in the cell nucleus, but they're also found in the cytoplasm. There's 3 types of RNA molecules: messenger RNA, transfer RNA, and ribosomal RNA. Now, a unique type of molecules that is an RNA nucleotide is something known as ATP. ATP is actually the immediate energy source for our body. When we take foodstuffs and break it down in process of metabolism, we break it down to generate these magical ATP molecules. Now, what are these ATP molecules? Well, they actually are a modified adenine nucleotide. an RNA adenine nucleotide, in that you're going to find the adenine nitrogenous base attached to a ribose sugar attached to that same phosphate. But notice what's unique about this phosphate. Notice that you have those two additional phosphate groups attached. And you can see those red zigzaggy lines there. Why are those lines there? Well, those are high energy bonds. And it took an enzyme and a lot of energy to create those bonds. Why are they high energy? Well, look at the molecule itself. Look at the phosphate. Notice it's a negative charged molecule. Imagine if you take two magnets that are both negatively charged and you try to push them together. Remember how they repel? Well, the same is going to happen here if I try to take one phosphate that's negatively charged and take another phosphate and put them together. They're going to have this energy, force energy here. And we take that to our advantage. We take enzymes and create the bonds, hold them there. So as soon as I need energy, at the split moment I need energy, all I have to do is go to the ATP molecule, chop off the last one. And I take the energy that comes from that energy that's sitting there, potential energy, and I break that bond and I take that energy to drive all the reactions that occur in my body. Well, now when you take that ATP and you break that one phosphate group off, what you get is ADP, which is adenosine diphosphate, an inorganic phosphate molecule. Now we'll enzymatically then use an enzyme called synthetase, and we'll actually put that phosphate group back on there enzymatically and create ATP, which is adenosine triphosphate. Now it's time for you to do a lot of reflections. We finished this whole episode of chemistry. We've looked at inorganic chemistry. We've looked at organic chemistry. We looked at the value of water, the value of the salts, acids, and the bases under inorganic. We also took a look under the organic of the four different types of organic molecules when we looked at carbohydrates, lipids, proteins, and now nucleic acids. It's your role now to try to put all these together and see the values of each one's of them. I'm going to challenge you, though, that throughout this semester, I'm going to want you to reflect back on this chemistry each time you're introduced to a new molecule, because it's going to be of value to you to determine whether that molecule is polar, nonpolar, whether it's a carbohydrate, a protein, a lipid, or nucleic acid, because it's going to have characteristics based upon what kind of chemical it is. You all have a great day. See you later.